So in this question, we have figure 3 shows how students set up equipment to investigate the effect of pH on the action of the enzyme pepsin on boiled egg white. So we're investigating the effect of pH on the enzyme pepsin, and egg white is our substrate. So we have our pH 1, 2, 3, 4, and 5. The pH solution and pepsin is in the test tube. And then we have a 40mm glass tube containing boiled egg white. The length of egg white in each of these tubes is measured after 24 hours. The results are shown in figure 4. So here we have a picture and a table that shows the length afterwards. So we can see that in pH 1, the length of the tube after 24 hours was 36mm. Remember at the top there it tells us it was 40mm to begin with. pH 2 has gone to 10mm, pH 3 it's gone to 24, pH 4 has gone to 28, and pH 5 the tube has stayed at 40mm. You can also see this in the pictures too, so pH 2 there is clearly the smallest out of all of them. In the first question, it says to state at which pH pepsin was most effective. So some people put pH 5 for this question. At pH 5, there was no change, so it stayed at 40. Therefore, the pepsin had not done anything. It had not affected the egg white at all. The pH it was most effective at is pH 2, where you can see here, where the pepsin enzyme has digested the most of the egg white, so pH 2. The next question, state how the pH of a solution can be measured. You should only give one answer. I'm just going to give you two because there are two options. You could either say using a pH probe. Remember that pH is lowercase p, capital H. The other acceptable answer that you could have given is a universal indicator. Litmus paper is not an acceptable answer because litmus paper only tells you whether a solution is alkali or acidic. It does not tell you its pH. So next question. A student noticed that the boiled egg white in some of the tubes also became thinner. Describe how the method could be improved to obtain a more accurate measure of how much boiled egg white was digested by the pepsin. So the student measured the length of the egg white. However, in the question, it says that the student noticed that the egg white also became thinner. So to obtain a more accurate result, rather than just measuring length or width, what the student could do is measure the mass of the egg white. And to get the second mark, what you need to say is measure the mass before and after the experiment. So measure mass of the egg white before and after the experiment. The reason why you'd want to measure it before and after is because then you can calculate the either the percentage change or the change in mass. Okay. The next one, student extended his experiment by replacing the boiled egg white with fat. Explain why there was no change in the length of the fat in the tube at any time of the different pH solutions containing pepsin. So a lot of people gained 1 mark for this. Pepsin is the enzyme, and at this time they've changed the substrate from egg white to fat. Pepsin does not digest fat, or pepsin does not work with fat. So pepsin does not digest fat. A lot of people got 1 mark for that. Now the second mark was for explaining why. So the question says to explain, so why doesn't pepsin digest fat? Fat is the incorrect substrate for the pepsin enzyme, so it will not fit into the active site of pepsin. So we'll just put that. Fat is the incorrect substrate to fit in the active site of pepsin. Okay, the last question here says, name the product formed when the protein in boiled egg white is broken down. So all that question is asking you, what is protein broken down into, or conversely, what are proteins made up of? And the answer is amino acids.